We will now move on to Thomas Konstantinovsky. And he's a PhD student in Gur Yaari's lab at Bar-Ilan University, Israel. And he will be talking for the next 20 minutes now about emergent compression theory and immunology for enhancing our understanding of immune repertoires. Looking forward.
Thank you so much, Victor, and hello, everyone. I'm very excited to share with you today my latest research work titled A Novel Approach to T-Cell Receptor Beta Chain Repertoire Encoding Using Lossless String Compression. That's a long title for you here, so you don't feel alone here. So before diving into the actual details and the implementation we show in this paper, I would like to take you through an example that will emphasize the motivation behind this work. So in our day-to-day routine, when working with AIRR-C, usually when we want to transfer those AIRR-C, those data sets between our colleagues, we tend to not send the raw data, obviously, because of its sheer size. Now, we go to one of the many compression algorithms provided to us, use them in order to compress the data, and only then send it to our colleagues. Now, the question I asked was, can we somehow represent a repertoire, represent this large list of sequences in a way that will allow us not only to compress the file, but also to directly derive inference from this compressed state. So in other words, can we somehow, in parentheses, hijack this compression methodology into our benefit to directly derive inference? And before looking actually at the implementation, it's worth mentioning the two flavors of data compression usually used in the different algorithms implemented. So we have lossy compression algorithms, which, as the name states, lose some information, and they guarantee us really, really good compression ratios, but they do not promise us one thing, and that's one important thing. They do not promise us exactly the same data that was compressed, that it would be returned to us when we apply the inverse compression. Now, on the other hand, we have lossless compression algorithms, which, although provide compression ratios a lot smaller than the lossy compression algorithms, but they do guarantee us that when we decompress the file, we get exactly the same data. And that's a super important fact, because imagine that you send data to your colleague and 2% of your data gets lost. Those 2% might be the signal that your colleague will try to find later. So obviously lossless was the way to go. And we had to choose, where do we start from? Like, what compression algorithm can we use? So we went and looked at the timeline of lossless compression algorithms. And we knew, in order to prove the concept of utilizing the compression algorithms for repertoire representation, we have to choose the most fundamental algorithms before we go and try anything modern and complex. So we looked at the timeline. We started all the way from Huffman coding, and we settled down on the LZ76 compression algorithm, invented by two brilliant Israeli scientists, Avraham Lempel and Lempel-Ziv. Unfortunately, we lost them in the last 3 months, but nonetheless, their contribution still keeps providing new research and ideas. So having this algorithm at hand, it's worth looking at an example so we can kind of get a feeling of what actually it produces and to better understand how we build our model upon this representation. So basically, given a sequence of nucleotides in our case, the LZ76 algorithm goes and reads the sequence from left to right. Each time it encounters a new pattern, it adds it to a dictionary. And this procedure keeps on until the algorithm encounters a sub-pattern it already saw, in which case the window of the LZ algorithm increases in size and it tries to capture a bigger pattern. And this procedure keeps on going continuously up until we reach the end of the sequence. After we reach the end, simply we use the dictionary to encode our original sequence, and we get the compressed representation of the sequence based on, again, the LZ76 algorithm. So having those subpatterns in hand, having encoded the sequence, what can we do with them? Like, what's the next stage? And the utility of those subpatterns was exactly what we presented in our methodology. So we take a list of TCR beta CDR3 sequences, and it's worth mentioning that the entire set of results that I will later show is based on TCR beta CDR3 sequences, but it's not limited to them. Basically, any sequence can be used to create the data structure I'm about to show, but it's worth mentioning we tested it on CDR3 sequences only. So taking those sequences, we can apply the LZ76 algorithm on those sequences, on that list of repertoires. And now comes the part where we ask, okay, so now we have all those sub-patterns from all those sequences. How can we provide a singular representation of this repertoire, of this huge list of sequences? So what we show is that there are two approaches to take in such in the path for answering this question. We can take a union of all those sub-patterns and create a bag of words-like representation. We call this the simple approach, as I will later explain, okay? So we can iterate over each sequence. We can derive a sub-pattern dictionary for each sequence, and then simply take the union and count all the unique appearances of those sub-patterns. This will result in a count vector, which is already a naive embedding of the repertoire. So I went from tens or hundreds of thousands of sequences into a simple, quite short numeric vector that encapsulates the information on the entire repertoire. This naive representation already shows how it can encapsulate information in repertoires. And as you can see in the figure in this current slide, you can see that when we took almost 2,000 repertoires from 3 different data sets of HIV in the infected individuals, the Emerson CMV data set, and almost 1,500 repertoires from a COVID data set, you can see that using those naive embeddings, we already see an apparent batch effect-like effect appearing when we project those vectors onto 2D. Now, it's important to know that the projection is based only on the union of the sub-patterns, only the sub-patterns that weren't common. But we see that there are sub-patterns that are unique to each dataset, to each condition, which by itself poses a really interesting question to analyze as to, okay, what are those sub-patterns? Why are they appearing only in those datasets, and how they unify those datasets from other datasets? Now, we neglected a very important fact or piece of information by using only the union. And some of you might already thought about it. We completely neglected the sequential relationships between the sub-patterns. We can all understand that even though CG might be an important sub-pattern, but where it appeared in the sequence by itself is already a piece of information that we cannot throw away. That's exactly why we propose the probabilistic approach, where we take those sub-patterns and their sequential order of appearance and combine them into a single data structure, which is a directed weighted graph. This graph is, again, a singular representation of the entire repertoire, encoding the dynamics and the statistical properties of the repertoire itself. So having this graph, okay, how can we even imagine such a graph growing when we insert sequences into it? So the first sequence inserted into a graph would be a simple path, which is not so surprising. But when we start inserting another sequence, we see that a common stem forms between the two sequences, which again, for most of us, isn't a big surprise, as we know most CDR3 amino acids in T cells start with the Cas. So we see the Cas stem forming between those two paths. But what's interesting is what happens when we keep adding sequences. We see gradually with each sequence the interdependence between the sub-patterns, and the fingerprint that forms, that depicts the repertoire, becomes more complex, adapting itself to that specific repertoire. And you can see that even with 45 sequences, right, out of 10,000, 100,000, we already get quite a complex and dense graph structure that encapsulates the information of this specific repertoire. So we presented in this paper 3 types of graphs. And the one that follows the examples I just showed you is a naive graph. And why is it naive? Because it simply uses the subpatterns as they are without any modification. And it's naive because it contains cycles. Those cycles are really crucial when looking at downstream tasks. They completely, they aren't in par with the actual repertoire dynamics we're expecting. They allow for behaviors such as traversing back up the graph, which we do not want. We know there cannot be a CDR3 sequence that would be a palindrome, right? So that's a behavior we cannot allow, but it has its benefits in certain use cases. So alternatively, we presented two other graph structures with some constraints that do respect the repertoire properties and the repertoire dynamics we strive to achieve. In case we are working with nucleotides, we presented the nucleotide double positional LZGraph. This graph is essentially the same as the naive graph, but each subpattern, each node is connected with a suffix that represents the reading frame and the absolute position of the subpattern in the source sequence. The same logic is applied to the amino acid case, in case we work with amino acids, where we don't need the reading frame. We just connect a suffix which indicates the absolute position in the sequence. So we have an understanding on what's an LZ graph. We saw how they are constructed using the LZ decomposition algorithm. So now I will show you some of the downstream tasks we were able to achieve directly inferring the LZ graph. So we showed that when taking an HIV dataset and using the feature vectors derived both from standard K-MER approaches, the most simple solution when we want to kind of encode sequences, and when using the LZ naive bag of words representation we saw in the beginning, as well as eigenvector centrality values derived from the graph, which are basically the feature vector we can query from the graph. And we see when you can look at the box plots, and you will see that when modeling a data set of HIV infected individuals, we were able to classify between sex, between male and females of those individuals with a much higher F1 score using the LZ derived features. When it's important to note that the computational cost of deriving those features is completely the same as deriving K-MERs, yet it achieves much better results. And you can also see by the red dashed line that when we took a second HIV dataset with samples that neither were included in the construction of the graph nor in the training of the model, you see that those features are also more robust than using just K-MERs. Now, we also provided a new idea for a diversity matrix. We call diversity index. We call our diversity index K1000, and it's fundamentally different than the traditional diversity indexes we are using right now when working with repertoires. So we are used to measure diversity in repertoires by inferring Hill curve-based diversity metrics, alpha diversity metrics. All of them, in a way, are completely dependent on sequence abundance in a repertoire. Whereas our approach, the K1000 diversity index, is dependent on the sequence dissimilarity. We try to leverage the dissimilarity and the sequence-based dynamics of the sequences in a repertoire in order to infer diversity. So I'm about to show you a figure, but before I will tell you the setup for the figure. And you can see in my current slide that we took 4 different data sets. We have, repertoires. We have a repertoire from the Emerson CMV dataset. We have a single repertoire from a COVID dataset. We have a random repertoire, which basically is a long list of random sequences. And we have a low information capacity repertoire, which in other words means that all sequences are unique and they look like real T-cell CDR3 repertoire sequences, but the change between them are up to a few nucleotides that are really similar to one another. And all of these 4 repertoires, they share both the same depth and the same alpha diversity. So basically, if we try to infer the diversity with our current metrics, they will all have the same diversity. That's the fact that we would conclude without using K1000. Now, the way we derive K1000 is sampling 1,000 unique sequences from each repertoire. We build a graph from those 1,000 sequences. We count the number of nodes, and we repeat this K times, averaging the amount of nodes we got with each sample. This applied on the 4 repertoires produces the following graph. Now, when we look at the average number of nodes we received when taking 1,000 sequences, which is the K1000, we see that even though using the traditional diversity metrics, all of them share the same diversity, you can see here how they are completely different from one another, where the random repertoire gives us an upper bound for what's a really diverse repertoire, meaning like all sequences are completely disconnected from one another, having no connection whatsoever. And the low information capacity one, which means basically most of the sequences share almost the same kind of base as we would see in, like, for example, in B cells after somatic hypermutation. We would see that because of mutations, we have a lot of unique sequences, but they stem from the same father sequence. But when we look at the CMV and the COVID repertoires, you see that, again, having equal alpha diversity, they have different diversity in terms of K1000. And we see that the CMV repertoire is more diverse than the COVID repertoire in this case. Now, this could be applied to much a finer inference. For example, we show that when we look at different age groups in the CMV Emerson dataset, we see that with each age group, the K1000 diversity index goes down, which is in part to our belief as for today that the immune system, the diversity in the immune system becomes less diverse with age, right? So that's the K1000 diversity index. We showed that we can also leverage the LZGraph in order to produce Pgen, to produce generation probabilities of sequences within a repertoire. And we compared the generation probability values we produce for sequences to ones of state-of-the-art models like Sonia. And if you consider Sonia uses actual genomic data, it requires the V and J alignment of the sequence in order to model the mechanistic system of the immune system to predict the Pgen and correct for all the different factors that are present in the immune system. It's amazing to see that we can produce the same thing with a fraction of computational time as compared to Sonia, but also without requiring any error-prone alignment steps, which, as we all know, isn't perfect, right? Then, based on the structure alone of the sequence, we can already derive a Pgen for each of our sequences that is already highly correlated to state-of-the-art models. Now, again, I will just remind you that all of this happens from a compressed file, right? We haven't done anything, we haven't aligned the sequences. And in cases where we have certain repertoires, maybe of different species, not Homo sapiens, alignment might not be even a choice for us. We might not even know the source genotype to align to. So we can already get generation probability values without needing anything except the sequences themselves. Now, slightly connecting to what Guillermo mentioned about synthetic sequences and generation, we also compared the ability of generating sequences using the LZGraph, which intuitively should sound really simple as we have those different sub-patterns and the positional information connected to them, and a really dense graph that we can just walk on. We can just take a walk on this graph and get a random sequence which should resemble and does resemble the statistical properties of its source repertoire. And that's exactly what we showed. We compared how the sequences generated by the LZGraphs, how they compare to sequences generated by Sonia. And you can see on the left panel that when comparing the distribution of Pgens of our generated sequences to the source repertoire and Sonia's generated sequences to the source repertoire, we both are really close to the actual source repertoire. But on the right panel, you can see that when we apply this analysis over 300 repertoires from the HIV datasets and measure the Jensen-Shannon distance from the source repertoire, we are actually a bit closer to the source repertoire than the sequences generated by Sonia. I have a few take-home notes to conclude this presentation. So LZGraphs are a really compact representation of repertoires. They strive to achieve the same compression ratios as regular zip files, but they allow us direct inference. Diversity can exist outside abundance-based measures such as Hill curves and entropy-based measures, and can be measured directly from the sequence structure. Many repertoire statistical properties can be directly inferred from compressed files and LZGraphs in particular. And this work, I hope, will open the door for more groups working and trying to incorporate the functions that lie behind compression algorithms as feature extractors and bases for deeper models in the future. Also, all of the methodology I presented here and the various downstream tasks, as well as many others that, again, due to the constraint of time, I didn't have the opportunity to show, are available in a Python library we developed implementing all of these that can be downloaded simply, and most of the functionality is plug and play. Provide your list of sequences and just ask for the inference you want. I thank you very much for your attention, and I would love to take questions.
Thanks, Thomas. Very nice talk. Are there questions? If not, I can just quickly start. So maybe I missed it, but so you mostly worked on TCR, but it also works on BCR, or?
Yeah, during our work in developing this model, we also in parallel started implementing it on full BCR sequences. Mm-hmm. We haven't taken this approach till the end due to the somatic hypermutation, and the inference becomes a bit more complex. The graph becomes a bit larger, but fundamentally the process is the same. The only factor that would change is the graph size and the computation time it will take to derive some of those things, but fundamentally it's the same thing. Yeah.
And then you showed this graph where you get this 0.82, I think, correlation with Sonia
yeah.
in terms of Pgen. So a bit maybe controversial question, but which do you think is closer to the ground truth, to the biological?
So it's a really good question, because intuitively one would think that a model that tries to resemble the biological process itself, right, taking into consideration self-selection, trying to model the probabilities of different VDJ events, right? One would think that actually leveraging and trying to imitate those will give you a lot more accurate Pgen. But at the same time, you see that only inferring the sequence level alone, you already get sufficient enough measure that compares itself to the entire mechanistic approach, let's call it, right? So I would pose a counter question, which would say, do we even need those, right? So it's quite simple to understand that, okay, if I'm a biologist, I'm interested in knowing like what VDJ event was specifically accountable for this specific Pgen. But again, even without mentioning that this requires us to know the alignment, right, which again is a problematic process by itself, this is not necessarily what we are looking for. And again, I would kind of side gear here when we're trying to understand the computational basis or like the mathematical fundamentals of the combinatorics and diversity of those sequences.
I guess that's a question.
Is it a high five or a question?
No, it's a question.
No, I was actually interested in this. So you also showed some examples where you learn these patterns for CMV and
sure,
HIV
yeah.
Repertoire, right? What do you think they kind of pick up on in terms of the kind of patterns you learn here, right? Have you thought about that? Like to what degree is it VDJ driven, what you find, and how is it kind of mixing up with the factor? Do you have any thoughts?
It's an excellent question, and we thought about it quite a lot, right? So just if we think about it for a moment, if we imagine a graph, right, especially when speaking about CDR3 sequences, it's kind of easy to imagine how the stem of this graph would be shared between many sequences, because most of them start with kind of the same stem. And most of them end with kind of a similar like triple or double F amino acid tail, okay? So the question is, okay, we know most variability happens somewhere in between, which is also accounted for the D gene somewhere hidden inside, right? But like, we look at motifs, and many papers mention the importance of motifs and how we can use motifs again to classify different things. But I think the LZGraph allows you to not only look at motifs, but also look at those subpatterns at their location and time, right? Like you incorporate some temporal information into this graph by considering, okay, so I saw TGC, right? So, or I saw ILP or some sequence of amino acids or nucleotides. But if I saw it twice or 3 times at the same position, and that's a characteristic of CMV, for example, right, or COVID, or and again, maybe the signal isn't singular, right? Maybe we have 15 motifs and we have all 3 or 4 sized combinations of them at certain positions. So those positions would be flagged with high probability. So when we derive the feature vectors, those subpatterns will get higher scores, basically, right? So I believe that's exactly what allows us to actually distinguish between different repertoires, and I would say even more encode and have like a fingerprint, right? A fingerprint for a repertoire in a shape of a graph.
Interesting, thanks.
Regarding this question, I think this was this plot where you had the 3 different immune states with all the small points. This, I don't know what the PCA or UMAP or I don't know. What was my question here? So you mentioned batch effects. Do you mean actually technological effects, or do you mean?
One would assume. One would assume. I would assume. Okay. So all of these repertoires are probably were sequenced with maybe different primers, maybe with different technologies, right? Which will already by itself would be some kind of confounding factor to distinguish between them. I'm sure that, like in most cases, one of the easiest tasks would be to classify a data set rather than to classify a certain aspect of a repertoire. Given 7 data sets, it's really easy to classify each data set, like by a certain, even noise, some background noise that could be appearing during the sequencing itself, that was all like persistent in all of the batch, and then reflected to us when we analyze the repertoire. But additionally,
So when you would learn on one repertoire and then generate new sequences, and if you were to, and if you were to do this for all of them, for all the different immune states, and then cluster them again, would they again cluster? But after the generation, would your generation?
So we have a figure in our paper showing exactly this. So we showed in our figure 3 variations of this exact plot. And we show that when we take only one data set, so we take only HIV, we build a graph from HIV, and we use that graph to encode all COVID and all CMV repertoires, right? So we have the same kind of base graph. We saw that we get exactly the same behavior, regardless of which data set we use as our base graph, right? And it goes back to this number, right? It goes back to, okay, there's this number of shared subpatterns between all of those sequences, right? We neglect those 2 sets of unique ones. The only thing that at the end of the day decides on how much those points would be away from each other is the relative frequency of those subpatterns in the graph, right?
Yeah, it's very interesting. Any more questions?
I have a quick question, just to follow up to the batch effects part. I do not know much about this. I'm just curious, how would batch effects be reflected in a typical repertoire data set? Is it at the gene usage or what?
I would doubt. Personally, I would doubt, and again, like it's only an opinion right now, not a fact. I would doubt that, like on the gene level, you would be able to see like a batch effect. But again, I would blame some errors maybe on the alignment step. But on the sequence level, if there's again a certain noise that maybe appear during sequences or some disturbance, and it's it was equal across the entire batch you sequence. So you sequenced a million sequences, and the noise is apparent in all those million sequences. That's something you would see in each data set, assuming each one had some specific noise to it during the sequencing or the procedure. But it's worth mentioning that the COVID one is made out of 3 data sets, right? So it's 3 different COVID data sets that were merged into one data set as part of the publication of like a large COVID data set. So even though all 3 of them were sequenced differently, right? If I were to color them by data set, they would be also like defined, like the red region would be also kind of separated. But in general, as a whole, because all of them kind of share the same stem, like which is COVID, right? The small noise makes some makes some distance between them, but they are still kind of following the same trajectory in terms of the 2D space.
Yeah, thanks.
About the batch effects, I would have think that the batch effect caused by primers may cause different gene usage. So
That's
I
a good
think
point.
it is possible to see gene usage because of batch effects. And also regarding your last sentence, then I think it would be really cool because the CMV is just a single data set, right?
Yeah. Mm-hmm.
So just enriching this graph with the other data sets,
Mm-hmm.
No matter what, and really be able to see better how
Mm-hmm.
different COVID data set more or less cluster together or assume the same trend of coordinates will be even more impressive.
Mm-hmm.
And it is impressive now.
Mm-hmm. It's a really nice suggestions. I really appreciate it. And if I may add a last sentence, we saw, like I showed here, that we were able, using the features, to classify between the sex of different individuals, which, as far as my knowledge goes, in different research papers we were looking for, this was not shown before. And regarding some of the points Gur mentioned in his talk, I wonder if in the path of selecting proper samples for different tasks, we can leverage somehow, better selection can be made if we can somehow estimate immunological age or like immunological gender based on a repertoire, in order to better direct our synthetic data towards our tasks, right? If you can generate synthetic data set and then classify it to be a female or a male data set, I think that's something that can be of use for us in the future.
Great. Maybe one last very quick answer, please. Since you come from a lab which likes to look at polymorphisms and all these kind of things, is there also a plan to incorporate this somehow in the modeling, since we now know recently that polymorphisms can change also how much gene usage and is there something that you got?
So we mentioned in the discussion that basically there's a really long list of things to do from here, even though there's some really nice results here. One of the biggest kind of goals of this paper is to open the door for our community to consider more approaches like taken from traditional computer science and not jumping maybe to really complex machine learning models straight away. So that's definitely a path to take, and it is considered, Victor. Thank you.
Great. So if there are no further questions, I think we can conclude today. Thanks again to both of the speakers, Gesche and Thomas, and looking forward to the next AIR seminar very soon. Thanks, everyone. Bye-bye.